Hi. So most bacteria can be broadly classified as either a gram-positive bacteria or a gram-negative bacteria. Gram-positive bacteria have a thick peptoglycan layer and no outer lipid membrane, while gram-negative bacteria have a thin peptoglycan layer and have an outer lipid membrane. Excuse me. This means that when the gram stain is applied to gram-positive bacteria, they stay purple because the crystal violet dye will stick to them and not be washed out, whereas gram-negative will either be red or pink because the crystal violet during the procedure is washed out and the seraphim is then dyes them a red or a pink color. This means gram-positive and gram-negative bacteria require different treatments, such as penicillin that will work really well on gram-positive bacteria because of the peptoglycan layer on the outside. Penicillin begins to break down and inhibit the production of peptid glycan, thus making the cells fragile and weak and almost leaky, like the stuff on the inside starts leaking out onto the outside. This makes the cell unable to survive, and thus antibiotics work on that. But they don't work as well on gram-negative bacteria because the peptid glycan is between the cell membranes, even though it's thin, and it is much harder for them to break down, so they need different kinds of treatment.